Okay, so hi. Um, just a second. Okay, so good evening, everybody. My name is Ankita. As you already know, we will be starting today's session. So just give me a moment. Yeah. Okay. So since nobody has joined yet, I'm going to go ahead and start the session for today, and it will be uploaded on YouTube for everybody else to study it as and when required. So basically, the topic we were covering in the last class, it revolved around the process of cell signaling, which usually takes place in cancer cells. Now, taking into consideration the fact that we discussed the basics and wrapped up a few more loose ends about cancer signaling pathways in the last class, I'll go ahead and revise that first, and then we'll move forward towards the discussions regarding therapeutic targeting of those signaling pathways in the upcoming slides. So, just to brush it up, basically what we discussed in just a second, yeah, in the last class, it was centered around the key concepts of cell signaling, whereby we had discussed about the basic properties, including specificity of cell signaling processes. We talked about amplification of the signal, which means that once a signal has been or a stimulus has been received by the cell, that a signaling pathway has to be activated. After that, signal amplification takes place within the cells, and then there can be momentary desensitization as and when required. And also another property that is usually found within cells is that multiple signaling pathways are actually able to communicate with each other and there is crosstalk. And based on that, the integrated signal is used to stimulate the activation of the concerned pathway. And therefore, the readout that comes out from such a pathway is also a cumulative effect of both of those. So these were some of the basic properties that we had discussed in the last class. Then we moved ahead and understood the different types of cell surface receptors. Now, among those different types, we have, first of all, gated ion channels, and we had discussed the differences between ion channels or just channels in general versus receptors. So, starting off from the fact that their functionality, their involvement in pathways is very different. The way in which they allow the passage or the movement of different components. When I say components, for example, when you talk about an ion channel, say for example, the calcium ion channel, which we discussed in detail in the last class. So compared to an ion channel, we discussed the fact that a receptor, in contrast, would have a lot more specificity and a lot more regulation in place to allow the movement of any molecule across the membrane. Furthermore, we had discussed that receptors can also be of different types, starting off from receptor enzymes, which involve the big class of RTKs, which we have been discussing, and serpentine receptors, which included many different, um, not many different, but majorly the example of which we have studied in the last class, which is about GPCRs or G protein coupled receptors. Then we also understood receptors that do not have intrinsic enzyme activity, and that included enzymes like Janus kinases or the JAK-STAT signaling pathway. We also talked about adhesion receptors, where we discussed about cadherins, and I cited the example of EMT being the example of one such process, which is involved and which is a necessary step that occurs when cancer progresses from uh, uh, like as a stage of cancer progression when it develops from a primary site of tumor, and then it metastasizes to form secondary tumors throughout different parts of the body. We also discussed steroid hormone receptor signaling in detail, whereby we understood how steroid receptors, first of all, their entry into the cell is something which distinguishes or which segregates them from all other types of signaling pathways or signaling receptors. So the fact that steroid receptors are able to mobilize and they're able to move into the cell, and they're able to do so because of the nature of the receptor itself, and how after not the steroid receptor, sorry, the steroid molecule itself is able to move inside the cell, and then upon binding with the receptor, the complex is able to localize inside the nucleus, And steroid receptors are very commonly found to be transcription factors, and therefore they have a DNA binding domain based on which they're able to bind to their recognition motifs present on the DNA. They are able to regulate transcription of whichever genes they are able to control. And then those genes undergo transcription and translation, and finally, the necessary gene product can then be synthesized. So these were the broad concepts that we discussed, and we also studied the steroid hormone receptor in more detail, how it moves into the cell. Like, for example, cortisol here binds to the intracellular receptor protein, and then it is able to move past the nuclear membrane as well through the nuclear pore, and then control transcription of the downstream genes. Then we had looked at GPCR signaling or G protein signal transduction in a lot more detail. We understood how there are multiple types of G proteins, GS, GI, GQ, and G1213, and how overall a very complex circuitry of this family of receptors is able to control very basic cellular pathways, which includes organismal homeostasis, embryonic development, gonadal development, learning, and memory. And this is just um, it's the most simplest way in which this can actually be seen. But if you study about this in a little more detail, you'll see that GPCRs are everywhere, and they're very crucial receptors involved in very simple sensory pathways, like, for example, taste, smell, sight, or vision in general. All of these involve GPCRs in one way or the other, and they're very crucial. In fact, if you must have studied about rods and cones at some point, like during your school days, we have studied about the eye and how rods and cones are the light sensitive cells, and they're necessary for our vision to actually be functional. And there also GPCRs play an important role. Then there are gustatory GPCRs, which are responsible for sensations of smell, or for taste. There are a separate group. So basically, there's a huge class, and they all have different sort of functions. And the way in which they function was something we studied. I'll come to it directly from the schema. So in this schematic, we had understood how GPCR signaling works. So you have a 7-pass transmembrane receptor, and when I say 7-pass, it means that it's a receptor that is passing, or that is traversing through the membrane 7 times. It is folded in such a way that it moves through the membrane 7 times. And once the necessary ligand has bound to the receptor, in this case it is epinephrine, just given as an example, then this receptor is able to trigger the movement of the GTP, the associated G protein, which has an alpha, beta, and gamma subunit, from which the alpha subunit is able to sort of move away from it. And after movement, it is able to bind to some downstream enzyme. In this case, the example given is adenylate cyclase or adenylyl cyclase. Then this actually activates, this activated GTPase is able to then activate adenylyl cyclase as well. That generates cyclic AMP from ATP, and that acts as a secondary messenger. So we talked about secondary messengers in some detail in the last class as well, I think. And then we understood how it is able to maintain that level, because when necessary, cyclic AMP can also be degraded by cyclic nucleotide phosphodiesterase, and this degradation is necessary to maintain a balance, so that there is so that the basal level of cyclic AMP within the cell can be maintained. And that is important, because if the basal level is disturbed, then an increase in cyclic AMP will not even be recognized by the cell. So the basal level is controlled, and then any disruption or any change from that can be recognized by the cell as the response to some sort of stimulus which has come in. So that is how the GPCR pathway works. Here, we had discussed very briefly about secondary messengers, example being calcium and cyclic AMP and diacylglycerol, all of these proteins, and they are coming into play as part of the cascade downstream of the activated G protein. And these intracellular messengers, whether the examples that I cited, they are secondary messengers, but primary messenger in this case would be the ones which are upstream of it, which are first conveying the information only to one protein, and then downstream of it, secondary messengers are relaying information to multiple proteins, or they are sort of branching out inside the cell. So that is how they basically work. Two of the common enzymes that are activated by G proteins. One is adenylyl cyclase, like I just mentioned, and the other sort of subtype involves phospholipase C. So phospholipase C was responsible. We had already discussed this in the previous class. It is involved in breaking down, as we talked about it. ASE basically means it's an enzyme, and phospholipase means it cuts or it somehow breaks down phospholipids, and it leaves the lipid inositol phospholipid into inositol 1,4,5 triphosphate, which is IP3, along with DAG, which again is a second messenger as well, and they both can then relay the information downstream of it. Then we had studied this in a little more detail, the phospholipase C branch, which I just mentioned. So here you can see DAG and IP3, both of them are getting released, so they're branching out from the system. And then DAG is able to trigger the activation of protein kinase C, which opens up some calcium channels, which allows the flux of flux basically means the flow of calcium into the cells, and that coordinates or that aligns with the other response elicited by IP3. So IP3 also triggers the release of calcium from the calcium stores which are present within the cell. In this case, that is the ER, and then that gets released into the cytoplasmic region, and then it is able to trigger the activation of calcium calmodulin complexes, which can then trigger the activation of downstream enzymes. So you don't need to know this much detail, but basically the topmost enzyme is able to trigger the release or trigger the production of secondary messengers like like IP3 and DAG, and they mediate the downstream effect of that signaling pathway. So that is basically what happens. And this was an example of the TB53 pathway. We did not go into too much detail about it, but basically what I discussed is that it's a guardian of the genome. It is responsible for mediating the normal cell cycle and normal cell physiology. And mutations or somatic mutations in TP53 is under most circumstances, the most common subtype of any cancer in general. Like if you see a huge percentage of all tumors, they usually carry some sort of TP53 mutation. And because it has such a major role to play within the cell, this mutation is often considered to be a driver mutation, but it has been well studied. Again, I hope you remember what driver versus what passenger mutation exactly means. And in this case, it means that it itself is an TP53 mutation by itself is an oncogenic event, which means it is cancer causing. Now, along with it, there are a huge number of mutations that have also been identified along with TP53 to be present in patients, and they are considered to be as passenger mutations, which means that along with TP53, they help in sort of aggravating the disease and making it a lot worse. And here we had seen the different ways in which TP53 causes cancer, leads to genomic instability, metabolic reprogramming, invasion of metastasis is affected, it mediates drug resistance, proteasomal activity is disrupted, etc. And this is pretty much where we had ended. Now, as part of today's session, like I'd mentioned in the last class, I discussed with you guys that I'll not be discussing other signaling pathways, even though there are a lot. Um, I can just mention the names to you. You can go through it at your own pace. If time serves us at the end of this course, we will try to come back to it because they are also equally, if not more important than the pathways we've already discussed. Like, for example, the Wnt beta-catenin pathway. We did not discuss the hippo signaling pathway, the YAPTAZ signaling pathway. So all of those are sort of left open-ended. But the basic idea or the basic structure, I hope everybody understands by now that you have some sort of a receiver molecule right at the top, which is able to sense the signal that is coming in. Then it relays the information that, okay, this stimulus was here, so now you need to elicit the response. Next, these are called the signal transducers. And finally, downstream of it, you have those proteins which are able to respond to the stimulus that has come in, and therefore they are known as effectors. So that is the basic workflow that happens that is present inside the cell. So while I've written here that types of signaling pathways will be continued, but that basically involved the recollection or the revision of what we've already discussed. But instead, we'll move a little bit ahead and look towards how therapeutic targeting is actually done for oncogenic signaling pathways. We will wrap this up by today. It's not a very detailed topic. It will be done pretty soon. After this, in the next class, I will be discussing cancer diagnosis, and I want to do that in a little more detail. We will start off, like you can see in the flyer as well. We'll start off with the traditional methods. You might already know a little bit about it, but we'll go into details about it because when you hear of traditional methods, you know, of course, there's surgery. You have heard of chemotherapy, but then what is chemotherapy? We don't know that in details, right? And how is it different from what we'll be discussing today? All of those details will be taken up in the next class. So we'll connect it with today's class. And just to remind everybody, the next class is on Friday, and it will again be from 5 p.m. instead of 5:30. It'll be from 5 p.m. I'll send a reminder. Don't worry. And I'm very sorry about the changes that have happened. I just had a lot of work that couldn't be postponed. Okay, coming back here. So the question is, okay, so we learned a lot about cancer cell signaling, basically oncogenic signaling. Now the question is, what do we do? We know that, okay, this happens. We understood that based on patient samples, scientists around the world are trying to figure out, okay, what all goes wrong. Now, what do you do with that information? Of course, the broad goal, the long-term goal in a bird's eye view is always to figure out a way to fight off cancer. Why do we say fight off? It's because despite all the effort that has gone into curing cancer for decades and decades and decades now, we always face some challenge or the other. That's because cancer is a disease, is extremely smart, and no matter what type of weapon you come out with, cancer has something better, and it always, you're one step behind. So that's why the approach that is taken towards cancer treatment is to try and mitigate the disease, which means that A, you try to stop the disease from progressing further. B, you try to minimize the disease. Basically, you don't want it to be spread everywhere, which will make its removal or the recovery process a lot more complicated. But instead, you localize it or you push it into one place. And third of all, you try to extend the duration of the patient's life while trying to conserve the quality of their life as well. You can't just, leave a person in a half dead state and then say, okay, yeah, that person lived for 20 years. No, that's not the point. That you want to give the best quality of life. You want to clinically manage the disease and you want to figure out a way to keep the disease from relapsing again as well. So in a lot of cases, cancer is removed, but then there's something or the other always happens, and in more cases than you would actually imagine, cancer does relapse and it comes back much worse, much stronger, and much more difficult to treat. So in that case, all people usually look at palliative treatment, which is sort of a management of the patient instead of looking for approaches to okay, cure the disease completely, but just to mitigate their symptoms, just to make it easier for them to manage day-to-day life. So just I wanted to give you this perspective of how the disease is actually looked at, uh, just to be a little more practical than just thinking that okay, I have a disease, I'm going to cure it. It's not that simple. It's a very complicated disease, and imagine we are not even learning 10% of it, not even learning 1% of it. We're learning just very, very, very small smidges about the disease. There's so much to know, so much to understand, and then so much to figure out and standardize for patients, like before it can actually go to patients. So a lot has to be done. Now, coming to the next slide. So, like I started off the discussion initially for today, how do we salvage the situation? We now know that a lot goes wrong in cancer. What can be done? So, here this is a representative comparison between the usual terms of therapy that are most commonly used. You must have heard about this at some point or another. Basically, the one which we are more familiar with, which we hear of more often, is chemotherapy. Then comes targeted therapy and then immunotherapy. So, in today's class, what we will be discussing in a lot of detail is the fact that what is targeted therapy? What does it mean? So, let's just quickly go through a glimpse through what these definitions exactly entail. And We will be discussing chemotherapy in the class where we talk about traditional methods of treatment, of cancer treatment, which will happen in, I guess, three classes most probably, and immunotherapy, which is the more advanced, more recent way of treating cancer, and which has a lot of ifs and buts associated with it as well. And that topic will actually blend with the concept of precision medicine. So we'll do it sometime around that time. Excuse me. Yeah. So, how does it work? First of all, what is chemotherapy? The basic idea, you want to target rapidly dividing cells. What is this concept based on? Like you remember, if we just go back to your first class where we talk about what is the definition of cancer, then the most common idea about the disease itself is that there is uncontrolled cell proliferation. That is what people think of immediately. So the most traditional method actually aimed at targeting or inhibiting this property, this most common property of cancer cell, which is that cancer cells divide. So to target the most rapidly dividing cells, people opt for chemotherapy. And what are its side effects? It causes hair loss. Very commonly, you must have seen it. And I think I discussed it in one of the initial classes as well. Why there is almost always hair loss in patients who undergo chemotherapy, and what is the reason behind it? Like the stem cell generation is lost, all those parts of your body which needs expensive radiation, where it needs actively dividing cells, they are compromised, and therefore people suffer from extensive hair loss almost throughout their body. Then there can be intestinal damage and also nausea, which is, of course, something that happens very commonly with cancer. Now, what are the limitations? So, one major limitation, which again is something we'll discuss towards the fag end of the course, is that cancer cells develop resistance to chemotherapy. So, if you remember, we have discussed this before. If you give cancer a treatment, cancer is going to figure a way out, and therefore, most commonly, what happens is that your cells become resistant to the treatment that you had initially tried to inhibit them with. Again, next comes the second approach, which we'll be discussing today, which is targeted therapy. Now, what exactly is targeted therapy? It means that cancer cells are getting targeted. So, just a sec. Okay, so basically, it targets those proteins. There's some issue with light, but okay. It targets those proteins which are necessary for growth of cancer cells, and it looks at the most common symptoms that are looked at are liver problems, diarrhea, skin, etc. Here also cells develop resistance. Um, this is a concept we'll not go into today, but basically it's a progress from the initial concept of chemotherapy. But again, it has its limitations as well, and I think that's one of the questions I've put in today's like concluding slide for key takeaways and questions. That what are the limitations associated with the targeted therapy approach, and like what can you do about it? And the third subtype is of immunotherapy, whereby the idea is to use a patient's own immune cells against cancer. And obviously, the most common side effect or off target effect of this sort of treatment or this treatment approach is that it leads to autoimmune effects, which means that the person's own immune system is hijacked. And more often than not, it leads to some sort of a negative or should I put it, like a non-specific response against that individual's own cells, and therefore they suffer from an autoimmune disorder. And while it looks very tempting, the fact of the matter is it's very expensive. It is tailored, which means that it is specific for the patient who is undergoing that treatment, but it is very expensive, and therefore it's not the best strategy to go ahead with. So these are the three strategies. When I say best, I mean in the sense that it's not very affordable, it's not very practical, but yeah, it seems very promising. And every day you can find some or other advancement or some or other progress in the strategies which are used for immunotherapy. But yeah, while it seems very promising, the practical application of this is quite difficult, and not everybody can go ahead with it. However, if you read a little bit about it, if you look at time lapses about the disease, you will see that if you look at the last, say, 50, 60 years, you'll see that over time, every treatment approach, which was initially, which seemed like almost unaffordable for everybody, over time it has progressed in a manner such that it has become affordable. For example, chemotherapy at one point of time, however much it used to cost, that has actually reduced over decades, over the different approaches, over pharmaceutical companies taking over. All of this has come into play. So you can expect that maybe 50 years from now, immunotherapy will become like a very affordable treatment, like how you take off-counter drugs and all of those. So medicines which are available just over the table, similar to that, you might be able to figure out a way to actually get immunotherapy that accessible for cancer patients. However, right now it's a far cry, and right now it's very unaffordable. But we are making do with it as of now. However, our focus today will be about targeted therapy. Now, what does targeted therapy exactly mean? So basically, the idea behind targeted therapy, why we are discussing it today, is because it actually looks at the concept that we have discussed for the last, I think, almost 2 weeks, which is about the signaling pathways which are operational in cancer. So if you remember, I had shown you like I think when we were ending the class from last week, then I had shown you a snippet of all the different possible approaches for targeting all of these signaling pathways, but we will look into that in more detail today, so don't worry. So, coming back to the definition of targeted therapy. Targeted therapies, the idea of these are made to find and attack specific areas or substances in cells, or they can in cancer cells, not just any cells, or they can detect and block certain kind of messages that are sent inside a cancer cell that tell it to grow. Basically, it will disrupt some sort of a growth stimulating pathway, or it will block that access. So any ligand which actually triggers growth of cancer cells, that signaling circuitry will be disrupted, or it can be in such a way that it is able to attack certain areas of cancer cells and avoid or blocks the communication of cuts off the communication between the two. Now, some of the substances in cancer cells that can become such targets, what kind of possible approaches they can be? First one is that too much of a certain protein, if it is present on a cancer cell. What do we mean by that? It basically means that on the cell surface, if you have some sort of a receptor which is very abundant. I'll give you an example, and we have a slide for that towards the end as well. In context of prostate cancer, for example, there is a certain protein known as PSMA, which is prostate specific membrane antigen. No need to remember, just learn about it for now. Basically, look at this, for example, this tumor antigen shown here in yellow. So imagine that this is a prostate cancer cell, and on the top of it, at the top of it, on the membrane, you have PSMA receptors, very abundant or present throughout the cell. So using an inhibitor or using a drug that actually binds to and that inhibits that particular target, example being PSMA, if it is possible to design something like that, if PSMA has a role to play downstream of it, then in such cases that will disrupt the entire circuitry. And therefore, if there is too much of that specific protein present on the cell surface, on the cancer cell's surface, then what it will do is it will eliminate non-specific binding to other types of cells. What do I mean by other types of cells? Most commonly, if you have a population, say for example, you have a tumor mass made up of, say, very vaguely speaking, say it is made up of 1 million cells. From that, if you have, say, 30,000 cells which are actually tumorigenic, and the rest of it seemingly are normal. So then what it will do, it will actually, the job of the treatment strategy is to actually only kill those cells which are tumorigenic in nature and not those which are healthy in nature, because they might have some sort of a useful function to play. So in that case, what it does is it selectively only targets those genes which are actually considered to be cancerous, only those cells which are considered to be cancerous, and not the entire tumor mass or tumor population. So that is the idea. Second, that actually forms the basis for the second point as well. That is for a protein which is present on the cancer cell, which is not present on the normal cell. So there should be mutual exclusivity between the two. What does mutual exclusivity exactly mean? Is that something which is present on cancer cells, not on normal cells, and vice versa. There is an exclusivity between the two. The third way in which these pathways or these strategies they can become cancerous, and as a result, they are very important. They're key strategies for targeting these signaling pathways as well. So one way is that a mutant or a changed protein can be present on the cancer cell. Genes or changes in the DNA that is not present in a normal cell, all of these can actually lead to or they can be considered as some key targets in cancer. If you don't understand anything at this point, there's nobody else. So, Ankit, if you don't understand, please let me know. I'll just try and explain it. It should be fairly simple because it's more example based. But still, if you have any doubts, let me know. So this schematic actually shows the same thing. So different strategies for targeted therapy have been shown here. One example of which is something I just discussed right now, which is whereby you have a tumor antigen which is present on the surface, and then you use a ligand targeted therapy to bind to that particular antigen and therefore affect its downstream mode of action. Uh, Yeah. Apart from that, you also have growth factor receptors. One example of which, if you remember, we had discussed about EGFR, which is epidermal growth factor receptor. And against these GFRs, you have therapeutic antibodies with or without toxins that are able to bind to the receptor. And because it is present in such high abundance on certain types of cancer cells, only in those cells the therapeutic antibodies or therapeutic vaccines will be able to work and they will be able to kill the cancer cell. Again, that is the broad idea. Then you have tyrosine kinase inhibitors like levetiracetam. You have apoptosis antagonists, angiogenesis inhibitors. Again, keep in mind why all of these different subcategories are present is basically because of the reason for which we have studied hallmarks of cancer. So you understood that there are many different parallelly operational pathways which are working inside cancer cells. And yeah, so targeting any or all of those can be a very good strategy. And if you remember, we had talked about how cell proliferations, the resistance of cell death, all of these are different attributes or different types of cancer cell hallmarks, among which angiogenesis was also a subtype. So for angiogenesis, inhibitors like angiostatin, endostatin, and avastin can be used. Apart from that, you also have metalloproteinase inhibitors and different strategies for immunotherapy, all of which we will look at uh, in some class or the other when we discuss either the traditional methods for treatment or when we discuss the more advanced or newer strategies for treatment or for controlling cancer cells, basically. Now, continuing with what we have already discussed, what exactly does targeted therapy mean? Some part of it we've already discussed, but apart from that, the action of targeted drugs can work in different ways. What exactly do we mean by these different ways? First off, um, think of it in a okay, my video is off. Yeah. So think of it as a basic idea. See, imagine that you have a cell. Now, your job is to you don't have one cell, you have a population of cells, one stained red which is cancerous and another, say for example green, which is non-cancerous, but that distinction is only possible because you have labeled them. Now when it is inside a tumor, when it is formed within a person's body, when it is formed in vivo, the situation is quite different. Why so? Because think of any 3D structure, think of a ball basically. Now, what can happen is that your drug might be present around that tumor because that drug is coming in solution. But cells which are towards the interior of that tumor, they might not actually receive the drug. Or even if they do, they might be receiving a very dilute concentration, as a result of which the efficiency of that drug working will be very different for cells which are towards the outside or cells which are at the core of the tumor or at the tumor core. So basically, the efficacy of the drug can depend upon what type of tumor it is, how abundant it is, and what percentage of the drug is actually able to enter into the cell. So this I just wanted to keep in mind because the reason I want you to understand this is because um, if you have worked in a lab before, in a molecular biology or a cancer biology lab before, you would know that the standard model, which is used for studying cancer cells in general and different ways of cancer therapy also, in that case, under most circumstances, we grow cells as a uh, like a sheet, like you have a mat or a sort of a monolayer of cells basically. However, that's not actually what happens inside a patient's body. Therefore, when you have cells which are sort of spread out on a surface and you're adding a drug on top, then obviously the drug is able to penetrate each and every cell. However, when the situation is different, whereby you have a mass, then tumor is able to block out the center of the or the core of the tumor is sort of blocked out. As a result, drug can only sit on or can access the cells on the outside and not towards the core of the tumor. This is one of the reasons that while you might it's difficult to optimize the dosage of these targeted therapy drugs. That is because if you even if you keep on increasing the dosage so that the complete eradication of cells can occur, that's not always possible. That's because at the core of the cancerous mass or at the core of the tumor, usually some cells which are known as cancer stem cells, they are present. And those cells, as you can understand from the concept of stem cells, they are capable of regenerating, and because they are considered to be cancer cells, that is why they are able to proliferate. When I say regenerate, I mean they are able to divide extensively, and they are the core of the I mean, they are the core reason as to why a tumor has so much variation. So basically, that is the reason why, despite the fact that sometimes drug dosage is increased, you're still not able to see decrease in tumor size or basically remission of the tumor. So one of the reasons is because of the structure. Therefore, for correctly studying how functional a drug may or may not be, one strategy involves the usage of different types of model systems for studying cancer. I will probably, I don't think it's a part of the flyer, but I'll probably discuss it uh, a little ahead, not right now, after we have discussed some other core concepts like treatment, diagnosis, all of those. Following that, we might discuss this. Now, the action of targeted drugs, how do they work? Despite the fact that, yes, there are a number of challenges, how do they exactly work? First of all, they are able to block or they are able to switch off chemical signals which tell the cancer cell to grow and to divide. Second, what does this mean? It means it blocks the signaling circuitry. When I say block, it basically means that there might be some protein X part of a particular cascade, and there is some sort of a targeted inhibitor. Uh, just think of anything, inhibitor A, and then that inhibitor is able to go and bind to that to the protein which is transcribed by gene X, and as a result, gene X mediated signaling is completely switched off. So nothing is mutated. It's just that the pathway is switched off and it is no longer responding to the presence of a ligand at the top. Second, targeted proteins or targeted drug strategies, they can often change proteins within the cancer cells, so the cells are actually dying. So what does this mean? It means that within the protein, the first one you looked at, that is blocking of chemical signals. More commonly, you will find these receptors to be on the cell surface. Second, the drug is modifying the intracellular protein in such a way so that cells are no longer able to work. Third is that you give a drug which prevents these cancer cells from forming new blood vessels, which means they are angiogenesis inhibitors, and fourth, you can carry toxins to the cancer cells to kill them, but not normal cells. So targeted drugs, you may ask that how exactly can you ensure that? If we could, then that would just solve the job. Well, there are strategies to do that. These approaches majorly use some sort of an antibody because antibodies are very specific to the protein against which they have been raised. And therefore, these antibodies can often carry a tag with them, and that tag is actually a toxin. So if you have, say, a protein X to be present in very high quantities on the surface of a certain cancer cell, um, then what can happen is that if the antibody is able to bind to that protein of interest, then what potentially can happen is that the toxin which is attached to that protein of interest will also be able to attack the cell, and then the cell will die. So that is the basic idea that is usually used for targeted cancer therapy. There's different strategies that can potentially occur. The action of the drugs can affect where these drugs work and what side effects they cause. And it's important to note that some targeted therapy drugs, for example, monoclonal antibodies, they work in more than one way to control cancer cells, and often they can also be considered as immunotherapy because they boost the immune system. So, this I will be discussing immunotherapy in further classes, so maybe we'll not go there right now. But I'll just show you some examples of some names of cancer drugs. Basically, if you see here, you look at different subclasses for cancer therapy, schematics of targets for cancer therapy. The first major class is growth factor receptors. You've already studied a lot about it and you have some idea about the way in which it works. Second, you have hormones, which are working via the steroid hormone receptor channel or a steroid hormone receptor mediated signaling. Third, you have other signaling pathways, which involves BRAF, mTOR, etc. And finally, you have some stimulus coming in from the tumor microenvironment, and different types of drugs can actually help in inhibition of those. So if you look at some of the examples for growth factor receptors, for example, you have trastuzumab, you have cetuximab, you have bevacizumab, uh, each of which are targeting different cell surface growth receptors. Um, yeah, for example, if you look at cetuximab, it works on EGFR. You have something else working on HER2, VEGF, etc. So these are how cancer therapy for growth factor receptors can actually be designed. For hormones, we have already discussed pretty much the same thing is being followed. For, for the tumor microenvironment, you can see that there are certain types of drugs which help in dissociation of the activated T cell, and yeah, then it can do its basic job. Now, one thing I would like everybody to note is that if you see, then a huge subsection of all of the different types of treatment strategies, um, like if you look at these names, you will see that they have MAB at the end. What does this MAB mean? MAB basically stands for uh, antibody, which is monoclonal antibody. So cetuximab is monoclonal antibody for EGFR, and it is used as a strategy for immunotherapy. So MAB basically stands for monoclonal antibody. That's what I wanted you to understand. So every time you stumble upon something like this, you would know. So we'll move ahead, but before that, Ankit, do you have any question regarding what we have discussed till now?
No, no.
No? Okay. Just give me a minute. I need to attend one call. I'll be back in 1 minute.
Okay.
Hello, am I audible?
Yes, ma'am.
Okay. So, coming back to this. So, we discussed discussed the basic definition of targeted therapy. Now, I'm going to give you some examples for this in more detail. So, the first pathway that we discussed when we talked about cancer signaling, that is exactly where we're going to start. So, for the basic approach for cancer signaling pathways, which obviously depends very heavily upon the ability of cancer cells to divide or to proliferate, came as the first most promising target. So basically, if cells divide, the cancer cells are able to divide the fastest, you would want to inhibit that. Now, the question is, how does that differ from what we had discussed as the definition of chemotherapy? So when you talk about chemotherapy, I will be discussing this in more detail, but those drugs, they do not target a signaling pathway. Instead, what do? They are inhibitors of certain processes which are crucial for cell division. Most commonly, cell division or cell cycle progression is what they use to interrupt. Um, Let's take an example. If you have studied about taxols or taxanes, if you've studied about cell division in detail, you would know that it basically interrupts the process of separation of two cells, two daughter cells, when they're undergoing cell division from metaphase to anaphase. When that separation or that microtubule assembly occurs during that process, that entire process is disrupted. So taxols are one group of drugs. It's a huge group, taxols or taxanes, and among those you have different types of taxols which are usually found. And this was a preliminary strategy which was used many, many years ago. And while it seemed very promising that, okay, you're just going to stop cells from dividing, the problem with that was, how are you going to ensure that you are not affecting other cells? So, for that purpose, people shifted from that and instead looked towards proliferation inhibitors or cell division inhibitors, but which are going to be more relevant for those cells which are undergoing active division. And that is better focused at when we talk about targeted therapies. Again, this is not like a foolproof situation, it has its own limitations, but we are going to learn everything that happens. And the reason we are talking about this in more detail is because these are very actively being explored in clinical trials. And it is something which is practically more easy to translate. When I say translate, it means that something which can be used for patients. While like immunotherapy and all the current advancements that are going on, they are very promising. They are less closer to clinical use as compared to these strategies. So coming back to the pathway, you have already studied about this in quite a lot of detail. We have learned about RTKs and we have learned how the downstream processes happen. Now, you might not be very familiar with every protein that is mentioned here. Don't worry, you would know some of them. For example, you know about mTOR, you know about AKT, you know about RAS, some other, this entire MAP kinase cascade, you're familiar with those, MEK, ERK, etc. So all of these you might be familiar with. But basically, what this is trying to show to you overall is that under physiological conditions, while ligands are able to bind to these receptor tyrosine kinases at the cell membrane, which is what we just discussed, and induce the autophosphorylation of the RTK's catalytic domains, which happens towards the cytoplasmic side or the intracellular side, and the activation of downstream effectors occurs. What sort of downstream effectors? For example, P85 release, activation of all of these. You don't need to go into all of these. The one you have already studied is that there is an activation of RAS, and then RAS triggers the activation of the downstream signal transducers like MEK and ERK. So what happens here is that you can see that at each step there is a different sort of inhibitor which is being explored. Example being cetuximab, which I just talked about like some time ago. What is it going to do? It's going to inhibit this binding of the ligand to its of the EGFR ligand, for example, EGF ligand to the EGFR motif. Second, there can be a drug like osimertinib, which is going to inhibit the autophosphorylation. You have copanlisib, which is going to inhibit P110. Or let's take another example. You have trastuzumab, which inhibits the binding of HER2 to HER2 receptor. I want you to go back and study this, which cancer is trastuzumab used for very extensively. And it has been studied. It's not something very new. It has been studied for a really long time now. And it seemed very promising initially, and it's under consideration at the moment. Let's just keep it at that. These are all FDA approved monoclonal antibodies, which are most of them are monoclonal antibodies, which are shown here. And what you can also often find are some small molecular inhibitors, which are also another subcomponent of targeted therapy. So if you think of targeted therapy strategies as an umbrella, these are the two strategies which are usually looked at. So HER2 binding to HER2 receptor is targeted by trastuzumab. It's autophosphorylation by neratinib. Then you have the RAS inhibition or RAS inhibition of RAS phosphorylation or RAS activation by AMG. You have binimetinib. You have dabrafenib. All of these are there. This is a separate line or a separate axis, which is the steroid hormone axis. We will be looking at that. Don't worry. We'll be looking at that in context of prostate cancer again, because it's something I'm more familiar with. We'll come to that in more detail towards the, I think, in the last or the second last slide. But basically, what happens there is that in the presence of estrogen, the estrogen receptor it dimerizes and it translocates into the nucleus where it activates pro-growth transcriptional programs, which is something you have already understood by now. So this is estrogen shown here in green. What you see above it is that testosterone, which is present, which is circulating within the system of an individual, that gets converted into estrogen, and that is facilitated by an enzyme known as aromatase. So therefore, inhibitors of aromatase will disrupt that process. Once disrupted, estrogen is able to move, diffuse through the membrane. See, this pictorial representation shows you pretty much that because of the nature of the compound, nature of the steroid, it is able to move across the membrane. Once it does so, it is able to bind to the estrogen receptor, which is ER, and this binding or the movement of the ER and estrogen complex is inhibited or is stopped by tamoxifen and fulvestrant. Again, this is one more thing I want you to just find out which cancer it is usually found to be. Okay, somebody's joining very late. Okay, no problem. So yeah, so I would like you to read about this. You might find a link between trastuzumab, tamoxifen, and fulvestrant. Which cancers are they being currently explored in? Because it'll help you. I've not mentioned it in today's class, so it'll help you figure out how different types of drugs are more relevant for different types of cancers. Just give it a Google search. You'll be able to find out the answer for that. Sorry. Yeah. Apart from that, here you see a spectrum all of the different sorry all of the different types of inhibitors which are known for these drugs. So apart from the ones which have been mentioned, targeting RTK pathways in cancer is something that is extensively done, like I mentioned initially, because it is a pathway that is crucial and that is so varied. When I say varied, it means that you don't have just one RTK present within the cell, right? You have a spectrum of those, all of which are triggering eventually pretty much the same thing, that it is aiding cell survival, cell proliferation, cell differentiation. So therefore, stopping that or blocking that process is something which is very widely studied and very widely used for treating cancer. So apart from the ones that I've discussed, you can see that there are other types of receptors which have also been shown. FLT3, RET receptor, FGFR, VEGFR, EGFR or ERB, c-KIT, PDGF, TAM, and other RTKs. So we'll just go through it. You don't have to memorize all of this. It's just for you, your understanding that different inhibitors are known. Again, also to clarify the fact that you will have certain receptors which certain types of drugs which can sort of how do I explain it? It has some promiscuity, but under most circumstances, if you're using a MAB drug, which is basically what you see here, all of these drugs, they are MABs, which means they are monoclonal antibodies. If you have those drugs, they are extremely specific and they're going to only target the pathway which they are meant for. So Again, I'll come to why this is relevant. But before that, let's go to the different subtypes. Let's look at them quickly. FLT3 ligand, you can see some drugs like sorafenib, sunitinib, etc. RET receptor. Again, one more thing I want you to see. While these are just schematic representations, you will see that the way in which the receptor has been even schematically represented is quite different for different types, and that's the reason. There's a reason for that. It's not just because it looks pretty, because it is the way the protein might be organized. So I want you to go back and look a little bit at it as well. That why are some proteins only shown to have this sort of a domain which receives the ligand on the outside, and the rest of it just appears as sticks, whereas others have some very organized little domains on the outside and on the inside as well. Just give it a look if you have time on hand. So what you can see here, RET also has its own receptor. Now, what I was talking about, the promiscuity part of it, you will find that some of these receptors like sorafenib, you will find it in multiple classes of receptors, and that shows some promiscuity. However, if there is a certain type of drug which is meant to be functional for only one of these pathways, that will be considered to be more specific. Let me give you some example. Say, for example, trastuzumab. You will not find it in the other types of inhibitors for other pathways, which means that it is only specific for inhibiting this EGFR receptor and the EGF binding to the EGFR receptor. Trastuzumab, cetuximab, all of these are responsible for inhibition of this pathway. Whereas you will see some of these drugs to be more specific. So while they will be considered to be as targeted treatment strategies, if you use it to treat a particular cell, there will be pros and cons to this process. Why so? On one hand, while you are ensuring that that drug, say for example, you are treating a patient with sorafenib, this the one that we were talking about, sorafenib. What is the advantage of it? You will figure out that okay, this person, if they have some very hyperactive action or signaling of FLT3 and this VEGFR and say PDGF, all three of these pathways are wrong in that are going wrong in that person, then well and good. Using sorafenib is going to be a wonderful strategy in that case, because it will block all these three pathways, and therefore cancer cells' ability to proliferate will be completely disrupted. Now, on the other hand, if you have a patient who does not have mutations or who does not have a disrupted circuitry of any of these receptors, but instead has a different effect on it, then despite the treatment, what will happen is that the cells will not die. Say, for example, if a person has a mutation in the EGFR pathway, but they are being treated with sorafenib because somebody just figured out that, okay, this person has an RTK problem and their RTK signaling is completely wrong, then using that drug is A, not going to be effective. B, what it can eventually lead to is that cells might learn to become resistant. Because what will happen is that in those cells, which are being treated with sorafenib, despite the fact that it is not going to have any effect on it, what it will do is it will try to drive up the signaling of other pathways which will help in cell proliferation. So instead of being a beneficial approach, it might actually do something worse for cells. So usually, what not even usually, what is emerging as a concept right now is that instead of blindly just treating patients with a certain class of drugs just because they are figured out to have some sort of a dysregulation, what is the approach that we are shifting towards is to go for more precision medicine, more individualized medicine. So what you would ideally want to do is that for a patient, you would do a basic testing of a few genes, of a few genetic markers to figure out what might potentially be wrong. Why is that good? A, it will help you figure out that, okay, if a patient has mutations in XYZ pathways, I will look for inhibitors of those pathways. B, how will that help? It will help in reducing the time that it takes for the drug treatment to be effective. C, it will not lead to any bypass resistance mechanisms coming in, because you will make sure that whatever. See, again, I don't want to inculcate the wrong information in you that just using a drug is going to lead to resistance. No, I'm saying it potentially can. There can be a wrong off-target effect. And also, what can happen is that if a patient is being treated with a drug X, nobody switches, no doctor will immediately switch them from drug X to drug Y. That treatment continues for a long period of time. That person will be subjected to treatment for at least 2, 3 months on an average. I'm just giving you a broad timeline. So that is enough duration for cells to actually develop mechanisms for bypassing that and to become even more lethal. So therefore, what is being preached now as a strategy, or what is being suggested now to make it a make cell survival, or sorry, to make patient survival much better, for better prognosis of patients, is to go for a preliminary screening. The problem with that is, while it sounds very idealistic, it's very difficult to do in practical life. It's not possible for, like, think about it in this way, that no healthy individual, say for example you, me, or any other person, is not going to go for a screening just for, just out of curiosity. Nobody does that under normal circumstances, especially not in countries like ours, in third world countries like India. So usually people will only do that when they have faced some sort of symptom. Now that A is already too late, and B, at that point of time, the priority becomes treatment instead of screening. So what we are discussing is an idealistic situation. It's what you learn in books, but what you have to practically implement in real life in clinics is very different. And therefore, what might usually be the approach is that, say, for example, um, uh, okay, let me give you the example of trastuzumab. Okay, no, let's yeah, okay. If somebody has breast cancer, there is a certain set of drugs that they will be usually subjected to. But it would be ideal for that patient to undergo some screening. It's not a very long process, but it's ideal, but not what is done practically. That's just what I wanted you to understand. It's always better to use a more targeted approach, but at the same time, a combinatorial approach might be more effective for some patients. So I hope I could make it clear. It was quite long, but I just wanted you to understand the pros and cons of using different types of drugs. And these are the current challenges. There is no single solution to it, but this is something that each and every clinician and scientists and researchers and like medical workers, healthcare workers, they're trying to figure out. So effectively, the idea is that there are a lot of inhibitors for RTK signaling that is available in the market, that is commercially available, and that is under study in clinical trials and is found to be very effective for a large number of cancers. And most commonly, the pathways that are inhibited include PI3K, AKT, mTOR signaling, protein kinase C signaling, RAS, RAF signaling, STAT signaling, MAP kinase, and ERK signaling. What is it supposed to do? It's supposed to inhibit or suppress cell proliferation, differentiation, survival, angiogenesis, which is the sprouting of new blood vessels, and it is supposed to stimulate cell death. These are all considered to be part of cancer treatments. I hope you've understood. If not, let me know. I might have complicated the concept that I said a while ago. If so, just let me know. Other like subtypes of targeting signaling pathways. This shows you how PI3K activity signaling is also targeted in cancer. So this is a subtype of RTK signaling itself, but yeah, it's not under the RAS RAFMEC ERK signaling cascade. That's why thought I'll show it to you separately. So what you can see here is that downstream of the initial RTK activation, you see there is activation of the entire PI3K cascade that we have discussed previously. And if you remember, this is where PTEN used to work. And I had discussed multiple times that mutation in PTEN, or PTEN loss, as it is commonly known, is one major oncogenic event. And the loss of this particular enzyme, or the uh, loss of function of this particular enzyme, is a major subtype of a large number of cancers. One example again being prostate cancer. Now, inhibitors that can be designed for this pathway can also operate at several sub-steps. Like, for example, the PI3K inhibitors will, of course, operate right at the top. Then you have inhibitors for AKT, you have inhibitors for mTOR, which can operate in both these, for both these complexes, mTOR2 or mTOR1. And these are called as rapalogs, which are inhibiting the action of mTOR downstream, like the This is inhibitor of mTOR itself, like it is inhibiting the assembly or the functionality of mTOR and its entire complex, whereas rapalogs work a little downstream of it and they inhibit the action of mTORC1 on its downstream target. So again, not much you need to remember. Basically, they are inhibitors that are operating at different steps. So a little more detail about that has been given in the figure on the right. So like you can see, mTORC1 and mTORC2 complexes have been shown separately, represented separately. Their downstream functions are different. So if you look at mTOR1, you'll see that it's involved in protein synthesis, in lipid synthesis, nucleotide synthesis, and autophagy. All three of these processes are necessary for normal cell division because you will need to synthesize proteins, lipids, and nucleotides when a cell is dividing from 1 to 2. And there's reduced autophagy. Apart from that, mTOR2, which you saw here, is involved in increased cell survival and increased cell proliferation. So there are some classes of drugs which are identified to inhibit this PI3K-AKT signaling pathway, and it's very crucial and it's very much understudied. Some Just pay attention to the fact that some class of drugs are able to only inhibit mTORC1, whereas some inhibit both. So examples of that include temsirolimus, everolimus, nabrapamycin, ridaforolimus. They all work for mTORC1 only. And you have for mTORC2 a different class of drugs. These are labeled separately. Um, One thing I want you to note here is that if you look carefully, none of them are labeled as MAB. They are not monoclonal antibodies. They are instead inhibitors. They are like chemical compounds. So most commonly, when you find names like this shown as CC223 or OSI027, most commonly it's not a hard and fast rule. They are used for small molecule inhibitors. Um, what are small molecule inhibitors? I want you to read a little bit about it, but just as the name basically suggests, they are small molecule compounds, chemically synthesized small molecule compounds, which are able to bind to some proteins of interest and they're able to inhibit their activity. And one of the major advantages of them is their small size. So they are taken up quite easily, but they have their own set of challenges as well because their specificity is something that cannot always be guaranteed. The duration till which they would be effective, that can also not be guaranteed because they're not designed to target anything very long term, but small molecule inhibitor mediated mitigation of cancer or cancer treatment is something that is extensively studied. It's not a very recent technique. Again, it's been over a decade, much longer than a decade actually, that SMIs have been studied, but they are another subtype of the targeted approach for cancer therapy, like I mentioned previously. Also, while we are at it, just pay some attention to the difference in composition between mTORC1 and mTORC2. It's majorly with respect to Raptor, Ras40, and instead there is Richter and Rotor, which are forming that complex. And just the assembly of those two is responsible for triggering the activation of different downstream pathways. This I just wanted you to notice it a little bit. Uh, Following this, we're just going to go over these two. Yeah, okay, not a lot to discuss for today then. Okay, so here I have given you two more examples of targeting signaling pathways. Basically, one example is of the thyroid cancer. Uh, Sure, I hope you're familiar with the thyroid cancer, thyroid gland itself, but if not, just to quickly just show it to you. So this is what the thyroid gland looks like, and of course, it is located right below the larynx or the voice box, which you have here, and it sits above the trachea. So you have two lobes basically, and from the front, this is what actually the thyroid gland does look like, and at the back of it, because it sits like think of it like it's sitting here on the trachea. So from the other side, this is what it looks like. So there are four very tiny glands. So those are called as the parathyroid glands, and that is basically what the morphology of the thyroid gland looks like. Now, this is one example of how MAP kinase signaling pathways are very commonly used as a therapeutic strategy for thyroid cancer. So, uh, what exactly happens there? You have already studied this in a lot of detail. So, the MAP kinase cascade, as we normally know of it, involves the same RTK mediated activation. And you can see that these RTKs have been shown in a more generic manner because these particular drugs, which are lapatinib and CDX3397, can inhibit any and all of them probably. So, you have FGFR, EGFR, RET, and PDGFR. So, it's found to inhibit or found to suppress the signaling mediated by all of these RTKs. So as a result of this inhibition, the entire downstream pathway will be shut down. Apart from that, you have inhibitors of sotorasib, which is an inhibitor of RAS. And if RAS gets inhibited, this is where you will shut down the rest of the pathway. So what you're seeing is if you have a flow chart, then at each step you are trying to figure out an inhibitor so that you can disrupt the pathway right wherever it is feasible, depending upon where things are going wrong. Then from RAS to BRAF conversion, that is inhibited by the first generation RAF inhibitor, like for example, encorafenib, dabrafenib, PLX420, 4720, vemurafenib, etc. Then you have pan-RAF inhibitors. What does the name mean? It means it can inhibit all types of RAF, ARAF, BRAF, CRAF, all three of them. And these are some of the examples for the same as well. If this is not where things are going wrong, if it is downstream of that, you can use a MEK inhibitor, which is a MAP kinase inhibitor, which is selumetinib, cobimetinib, binimetinib, etc. Downstream of it, you have ERG inhibitors like olixatinib and this SCH compound. All of this just to iterate the fact that at each step, inhibitors have been synthesized, have been designed, and the objective behind that is to have a multi-pronged approach which is able to inhibit this pathway. Why? Because it is that crucial for thyroid cancer to develop, not just thyroid cancer, any cancer to develop. But this example was just given because it is something found to be quite abundant in context of thyroid cancer and is considered to be a very effective strategy. Coming to the last slide for today. So, okay. So therapeutic targeting of prostate cancer. Now, why did I come to that? Obviously, like I've said before, because it's something I'm very familiar with, and you will find an excellent example of steroid hormone signaling gone wrong. So let us first understand what happens in the disease. So let's zoom into this a little bit, maybe. So it might be a little hazy. I think, yeah, it's a little hazy. But okay, let's take it one at a time. So basically what happens there, you have androgen and the action of androgen receptor within the prostate gland. So what happens there? Much like the normal steroid hormone axis that you can see, the same thing happens here, that testosterone, it binds to SHBG. What is SHBG? Steroid hormone binding globulin. What is globulin? It's just a protein. It serves as a um, like a protective entity which is present bound to that particular testosterone while it is circulating in blood. From blood, when it enters into the prostate cancer cell or prostate cell for that matter, because androgens are required for normal function of the prostate also, then what happens? It enters into the cytoplasm and then it is converted into DHT, which is dihydroxytestosterone, which is the more active form of the same hormone. Then what it is able to do is that it can bind to androgen receptor, and then the androgen receptor can move into the nucleus. It will dimerize and then it will assemble with a few other parts of the some other proteins, let's put it at that, some other proteins, all of which will form a complex, and all together they will drive the transcription of several downstream genes which are very key, very core drivers or very key drivers of oncogenicity for that particular gland. Example being ES8 and PRIS2, and it will drive this increased growth and survival of prostate cancer when the disease is when there is onset of the disease. Now, under that, uh, with this basic pipeline in mind, which is pretty much what we have learned for any steroid hormone for that example, uh, much like that, we need to now understand what goes wrong, what types of things can go wrong, and at what step can we inhibit this process so that drug treatment can be made feasible. Why is this important? That is because this androgen signaling axis that I just explained is a major, like I can't even explain how like, you'll have at least 50 to 60% of patients who have some or the other mutation in this pathway. Now, 50 or 60% might look like a very small statistic, but think of it in this way, that if there are 10 patients for a particular disease and you're able to use the same drug for at least five of them and or six of them and they get better, that's a huge statistic compared to how cancer is leading to patient death worldwide, and people are just not able to fight off that disease. So any mutation or any change which is very prevalent in a population, which is very targetable for a population, is always desirable. However, it comes with its own set of challenges, that you cannot always use the same drug for the same population, because not everybody will have the same mutation. But when something is very frequent, it is easier to treat and it is easier to figure out. So, coming back to this particular pathway, so what exactly happens there? You've already seen that there's a lot that happens here. Okay, let's look at it step by step. Don't get intimidated by the figure. It has a lot of information, but it's composite, so I kept it here. So basically, under the androgen signaling axis, something that happens is this is actually how androgens are synthesized inside cells. So basically, different you have a starting molecule which is known as pregnenolone, and from there you have different sort of derivatives of that particular steroid. That involves progesterone. You have 7 alpha hydroxypregnenolone. You have development of cortisol from that. You have dehydroepiandrosterone, which is DHA androstenedione, and finally you have testosterone or dihydroxytestosterone. All of these are different types of androgens. So androgen is a broader term used for a set of steroid compounds which have a certain common structure. You can read up about it. But basically what happens is that there is a lot of conversion that happens among these. If you see that there's from the core molecule, there's diversion into two, from there there's conversion, all of these different substeps are involved to synthesize the final compound, which is testosterone, which is the main driver of androgen signaling axis in prostate cancer cells. So, what scientists have over time realized is that if we stop this conversion, if we stop the formation of the ligand itself, which is able to bind to the steroid hormone receptor, then that's a wonderful way to actually figure out a way to mitigate the disease. So, they have designed inhibitors for these enzymes which help in conversion or interconversion of these steroids, starting off with something like abiraterone, which inhibits the first conversion from the core molecule to the downstream molecules, following which you can see other inhibitors which operate at the substeps below it, or the conversion of, say, for example, let me show it to you like this. See, for example, the first drug, yeah. So the first drug you see here is abiraterone, which is involved in the first conversion from pregnenolone to the 7-alpha hydroxy compound. That same drug can also inhibit the step right beneath it as well. So in this case, repurposing or usage of a drug which is able to inhibit multiple steps of the same process is actually a smarter strategy. It would be very useful, and it will actually cut off the axis at the very top. So it is easier, or not easier, it's actually mm, a more effective strategy to stop the signaling downstream of it. However, it also has its own set of problems. It also has resistance associated with it. A lot of things have happened over time. But that is the basic strategy which is used. Apart from this, what can be done? In case you are not using an inhibitor like this and this ligand is getting synthesized, like DHT, for example, or testosterone, for example, which is the ligand. If that is synthesized, then what else can be done? There are a separate set of um, drugs which are often used. We'll come to that. What does it involve? One major problem that is often faced in prostate cancer and any cancer in general, if you remember, we have discussed this in previous classes as well, is that there is increase in the production of a particular receptor. If you remember, we had talked about the fact that there is amplification of RTKs and they are expressed at very high levels at the cell surface. I'll show that slide to you because that was a while back. Yeah. This is where we had talked about it, that there is overexpression or there can be amplification. I think it was there in the next slide as well. Yeah, so there's gene amplification, and so more and more receptors are expressed at the cell surface. So much like that, the same thing is happening here as well. The only difference being that instead of it being a cell surface receptor, it is actually a intracellular or a steroid hormone receptor. So there can be amplification of the receptor itself. So to inhibit that, what is often done is that a set of compounds can be used. One of the things that can actually be done is the use of some inhibitors like enzalutamide, apalutamide, darolutamide, all of these. No need to remember all of these. These are also just small compounds, small chemical compounds. They are not monoclonal antibodies which are used for this purpose. What sort of inhibition can they carry out? They can stop, they can bind to this AR protein or the receptor, and what they can do is they can prevent it from moving into the nucleus and then carrying out the transcriptional activity it was supposed to carry out. So they are going to stop the signaling axis from proceeding any further. Apart from that, they are also able to bind to mutant versions of the protein. See, WT indicates it's the wild type or the most canonical or normal form of the protein, whereas the others show you different sort of mutations. If you remember, we had discussed that in the first base, the first letter indicates the amino acid that it initially was. For example, threonine. 2A means it is converted into alanine. H means from histidine, it has been converted into tyrosine, leucine to histidine, or phenylalanine to leucine. All of these different conversions have happened. So such drugs have found to be effective for all these mutations as well, and they're able to inhibit the same. What is the reason for this? That is because, like I explained before also, that if you treat any cancer cell with a certain type of drug, cells are going to come up with multiple ways to bypass it. So these are found to be some very common strategies which are often found to be operational in prostate cancer cells to bypass any sort of treatment. So normally what is found is that AR is found to be very amplified, or AR can carry different types of mutations, or AR can be synthesized as a partial protein. What is a partial protein? So, if you remember, we had discussed this as well, that a incomplete protein was synthesized for RTKs where only the tyrosine kinase domain was present. So, analogous to that, instead of a receptor, androgen receptor being synthesized with both the ligand binding and the DNA binding domain, instead what is synthesized is one which only has a DNA binding domain present. So that in itself is independent of any ligand binding, and it can just do its job. It is capable of functioning by itself. So this is what you see is an incomplete protein. So such a protein which only has the DNA binding domain can move into the nucleus and then it can carry out transcription. You can see it is green in color and it is able to do its job. It does not require any ligand binding. So some inhibitors have been synthesized again only this as well. Example being the N-terminal domain inhibitor EPI or niclosamide. They are also able to target it. Apart from that, we also have some inhibitors which are synthesized for GR, which is an analog of androgen receptor. So if you remember, we had discussed this promiscuous activity to be present for steroid hormone receptors as well. So AR and GR, they are quite similar to each other, they're quite homologous to each other. And in some cases, when you inhibit the function of AR in a cell, GR takes over. So these are just 10 different strategies that cancer cells are going to use just to bypass treatment and survive irrespective of what condition you gave them. So what has been designed over time are drugs which are able to inhibit all of these different pathways. So somebody must have started off with just using a drug like abiraterone, which is shown here right at the top, that you stop synthesis. So then what the cells started to do is that, okay, they will synthesize the protein normally, but instead they will make the ligand much more in number, so they'll be responsive much faster. Then cells started to make an AR protein which lacks the ligand binding domain itself. So over time, as people have faced challenges, they have designed a class of drugs which can be used for different sorts of treatment. So these are just some examples I wanted to show you. So apart from these strategies, two other strategies that I also wanted to highlight for you. One was BET inhibition. So BET inhibition, without going into too much detail, what it basically does is uh, it stops the function of the entire complex which is required for transcription. So till now, what we have just seen that these are steroid hormone receptors which move into the nucleus and then they're able to transcribe. But they cannot transcribe by themselves. They have an entire ternary complex that also needs to be present, and all together that helps in transcription to be facilitated. So that complex formation and that transcriptional activity is stopped by BET inhibitors. And apart from that, there are some antisense oligonucleotides also. Not something you need to learn about, but basically it binds to the androgen receptors DNA binding domain and does not allow it to bind to the actual DNA and inhibits its activity. So these are all the different types of cancer signaling. Like I wanted to give you a flavor of different types of cancer signaling pathways and different types of druggable strategies that can be used. Starting from the most obvious one, which we started off with RTK inhibition using monoclonal antibodies, and that too of many different types. We looked at like a cascade of them, some of which have promiscuous activity. Then we looked at PI3K-AKT inhibitors, the rapalogs, and mTOR inhibitors. Then we looked at the class of drugs which can be used for steroid hormone receptors and how drugs have been designed for different types of approaches and how different types of um, different types of cancer cell modifications can be taken care of by these class of drugs, and there also inhibition is facilitated. So overall, that is the axis I wanted to explain to you. So I might have gone by this part really fast. So I think we are done with the material for today. I had expected questions, so I had designed a smaller PPT for today. So any questions, anything you want me to repeat, please unmute and let me know. We'll get to that before going into the summary and takeaways for this class. Any queries, anybody?
Actually, ma'am, this was the first time first time I'm seeing all this. Like, never read about this before. So
Mm-hmm.
to read the PPT once, and then in the next class, I will ask you questions like if I would have any doubt. Then
Okay, okay, yeah, sure, no problem. But did you understand the flow of it? This was just nothing for you to memorize or nothing for you to retain very strongly, just to understand that there are different ways of targeting signaling pathways. That is what I wanted to communicate with you. Rest of it, it's all, see, the more you get this, I'm just showing you in more detail because this is the cancer I'm more familiar with. If you look at breast cancer, there's a whole other axis that is targeted there. I just showed you one flavor of it. Okay, I'm telling you that already. I showed you the HER2 receptor, I showed you the EGFR receptor. So there are classifications in each type of cancer and details are extensive. The idea is just to understand that how you can use different types of drugs and how different substeps are inhibited in each cancer. So was that were you able to understand that? That's what I just want to know.
understood it.
Okay. Can't see the other person's name, but I think you joined pretty late, but that's okay. You'll get the recording anyway. But yeah, we will not have any more time shifts tomorrow. The class for next class will be for Friday, and after that we'll resume our normal classes Monday, Thursday, 5, 7 p.m. because it leads to a lot of disruption. I understand that everybody has their own schedules also, so we'll stick to that. And we will not go into too much detail about signaling from now on. We are wrapping up signaling for today. Any questions, if anybody has, of course, we'll take that up. And I think somebody had mentioned some of the preliminary questions I had asked in the first or second class about autophagy and all of those things. So I thought I'll take up all of the questions right at the end of the course for the last class. I think it is scheduled on 31st, if I'm not wrong. So we'll have like an entire we'll do a recap of everything in 1 hour and we'll wrap up all of the questions that we have discussed as part of the course in that class. So that'll be easier. It'll just to refresh everybody's memory for the entire course. So that was what I had planned. But yeah, anybody has their own questions, of course, you can bring it up at any point of time. So we'll wrap up the session for today. So basically, key takeaways. First thing you need to remember, there are multiple signaling cascades that are working in concordance with each other. That is necessary to maintain the physiological homeostasis of any normal cell. Targeted therapy has become an advancement of the conventional approaches for cancer treatment. Targeted inhibition of cancer signaling can differ for different cancer types. I really wanted to stress on this fact because that's why I showed you examples of different types of cancer signaling. Like, for example, RTK inhibition, while it is used for prostate cancer, it's not the first line of treatment. The most common treatment used is inhibition of the androgen signaling axis. Now, you may ask why. Those cells are also dividing, these cells are also dividing. No, that's not the concept you need to understand. What you need to understand, why are those cells dividing? What is driving division in those subtype of cells? For certain types, for example, when we talked about thyroid cancer, the MAP kinase cascade is the major driver. The aberrant functioning or the dysregulation of MAP kinase cascade is present extensively in thyroid cancer cells or in other cancers. I'm just giving thyroid as an example. As a result, inhibitors of RTK, inhibitors of MAP kinase cascade would be very effective in that particular pathway. However, in cancers which are hormone driven, example being breast, prostate, or ovarian cancer, all of these types of cancers, it is more logical to go for inhibition of these steroid signaling axis instead of just going for RTK inhibition. They might also be effective, but if you disrupt the signaling cascade which drives cancer cell division or cancer cell proliferation in those subtypes, that will be more effective. So that is why, how do you figure this out then? What type of treatment is going to be effective for what type of cancer? You have to study the data from patients. Not you, I mean, by you, I mean scientists in general. So if you remember that paper where I had shown that entire schematic of TCGA or the Cancer Genome Atlas, just, we'll just brush up everybody's memory very quickly. Um, okay, I don't see that slide. Yeah, this one. So yeah, we had seen all of these different types of mutations, and I think yeah, I was present in that other PPT I had. So if you just go back to that PPT, you will recollect that there is a difference in all of these different sort of in the frequency of mutations which can occur in different sorts of pathways. And a screenshot. Just a minute. So yeah, so you will, that is how you exactly figure out what is going wrong in a patient. And from there, you will statistically figure out that, okay, if this is the prevalence of a particular mutation or a particular um, change to be present in a population that suffers from a certain type of cancer, then the treatment strategies will be designed accordingly. So that is the basic idea. Also, what you need to understand, apart from the fact that the type of cancer also dictates the type of treatment that is to be used, one more thing that also should be understood is that the population in which a particular cancer is occurring can also often determine what type of drugs can be used. Why so? Um, Say, for example, if we are only talking about, again, the same example, say we are talking about prostate cancer, the frequency of mutations within the population that suffers from prostate cancer in India versus that which suffers from the same disease in Africa, in say Caucasian cohort. By Caucasian cohort, you mean the Western cohort, the white population, them or the Chinese population. All of these different populations, they have different frequencies of different mutations. Why does this happen? You may ask. The logic behind it is that um, the lineage from which we are all derived. For example, in our case, we have some sort of link or some derivation has occurred from the African cohort or from the African population. So we have some similarity. The South Asian cohort has some similarity to the African cohort. However, the Caucasian cohort is very different. So depending upon the race or depending upon the subpopulation which is being studied, based on that, many a times the type of mutation which might be present often varies, and therefore the type of drugs that might be more commonly used for a certain disease can differ based on the region from which the person suffers from the disease. Just without complicating it, simply what it means is that A, your drug depends on what type of disease it is. B, it depends upon what type of mutation it is targeting, what type of change is present in that sort of cancer. And third, where is the where is the population from which that cancer has originated. All three of these factors should be taken into account, and yeah, that is going to drive the basic idea of what treatment approach needs to be followed. Apart from this, disruption of the oncogenic signaling circuitry is often combined with other therapeutic approaches for multi-pronged targeting of cancer cells. Multi-pronged basically means like you think of a fork, basically. So it has four prongs. So when you have the circuitry disruption, it is often used as a combination treatment with other strategies also, so that the treatment is more effective and recovery or fighting of cancer can happen at a much amplified pace, and therefore recovery can be much faster. That's the idea behind it. So these are some of the key takeaways. Rest of it is majorly for like jargon you need to memorize, so that's why I did not put it as a takeaway. It's something for your knowledge, not for your memory. It's not possible for you to memorize. You just need to understand the basic concept. What you should try to figure out that while we are calling it targeted therapy, does it have any side effects? Is it foolproof? And if it is foolproof, why has it not cured cancer? So read a little bit about other strategies for targeted therapy as well, because that will explain the limitations to you. Then read about FDA approved targeted therapy approaches. I showed you some, the slide where we showed the monoclonal antibody consolidated in one schematic that had FDA approved drugs. It's present in the legend as well, so you'll be able to figure out from there. And if you can, if it is possible for you, again, this is not compulsory and it's something we'll discuss eventually also, is that are there any ongoing clinical trials about it? I will introduce clinical trials to you towards the fag end of the course when we have like hazard ratios and all of those for discussion within the flyer. If you see, it's mentioned. So I will just briefly let you know what are the different stages of clinical trials. Why that is important is because to us it looks very idealistic that okay, I have figured out this X protein to be a problem. I have a drug Y for it, and okay, cancer is cured. No, that's not how it happens. And there are stages through which a drug has to go. A lot of verification has to be made before anything can be actually tried for a human being, because of course human life is prioritized. And you might be familiar with some like jargon and some terminology because of COVID. We all learned a little bit about clinical trials at that time. So we're just going to touch upon that topic as well. But what you can see, you can look at it if it's interesting to you. If you want to go into pharmacology or you're interested in cancer therapeutics, etc., you can look at it. And yeah, this is a question I'd asked in the previous class also, so I've kept it in continuity because we are learning a little bit more about it now. So if you were to improve the specificity and the efficiency of these drugs, what would you do? So while it looks like a good approach, it's not foolproof, like I just said. And once you know about its challenges, you will be able to figure out what you can do to make it better. So, yeah, with this, I'm going to end today's class. It is 7:15. Okay, we are ending a little bit early, but then there was not a lot that was, there was not a lot of interaction in today's class. So I think we wrapped up a little early. Yeah, Ankit, go ahead.
Ma'am, actually, I have a question. It's not
Mm-hmm,
regarding to the related to the class.
mm-hmm,
Where can I find head and neck cancer data
mm-hmm,
apart from the TCGA website? Actually, I am an MSc dissertation student in University of Hyderabad, and
mm-hmm,
my project is on head and neck cancer. So
mm-hmm,
the data, I had an idea that I can like it's a bioinformatics lab. So
mm-hmm,
I do the gene regulation network of the HPV positive and HPV negative head and neck cancer. So I
mm-hmm,
had an idea that I can like analyze this
mm-hmm,
gene regulatory pathway, gene
mm-hmm,
regulatory network on the basis of male and female. Right now, I'm doing like I'm dividing it on the basis of HPV positive and HPV negative HNSCC. But
mm-hmm,
I Like I'm thinking of also dividing it on the basis of male and female or on the stages. So, but I
Mhm.
didn't find enough data on the TCGA. So, is there any other database
Okay,
where I can
so
get this?
okay, so a common repository for retrieving this sort of publicly available data is CBio portal. So have you accessed that already?
No, ma'am. No, ma'am.
Okay, so you can just check CBio portal. If you go to that website, it gives you a combination of any data which is publicly available basically. So there you'll find subtypes, like you'll find a lot of categories to look at the type of cancer. Among that, what information you need mRNA expression data, mutation data, all of that you will get. And what you can otherwise do if you just want if you want to consolidate the data yourself, what you can do is you can go to PubMed and you have to use keywords and then search for it. You have to go for head and neck cancer, patient data, etc. And then you have to see who all has available data. One more way to go about it is to go to Geo2R, which is an NCBI repository, like it is owned by NCBI. It's called Geo numerical 2R. So you can go there and you can search for head and neck cancer patient data. So there, but then what you get there is actually raw data. You don't get analyzed data, you'll get RNA expression or ChIP-seq data or some other form of data, mutation data, whatever is there, that you can download and analyze as per your need. But pre-analyzed data is best to access it via CBio portal. It's very user friendly. You will get graphs and everything. So it's a good way to start, I think, at least.
Okay, ma'am. Thank you.
No problem. Uh, the other candidate, any question? Your name is not visible. Any questions? Okay. If not, then I'll end the class for today. Yeah. Did you unmute? You have something to say? It's not audible. But okay, thank you for joining. I'll wrap up the class for today. We'll again meet up on Friday and we'll move into approaches for cancer diagnosis, the traditional ones. And if possible, we'll try to blend into the current strategies for cancer diagnosis as well. I'll at least introduce that. And then we'll move towards treatment. And we'll have to wrap up the course by the end of this month. So I'll pick up the pace a little bit, but I'll try to minimize unnecessary details as well, jargon or something that is just to memorize. I'll try to omit that. So we'll do it in time, don't worry. Okay, so thank you for joining. I'll see you again on Friday. So have a good evening and bye.
Thank you, ma'am.